Now looking at the ultrastructure of the chromosome. Uh, when they study in the structure in the electron microscope, they can see that they are very fine fibrils, okay, very fine to say strands, okay, having the thickness of 2 nanometer to 4 nanometer. And since the DNA is 2 nanometer wide, so a single fibril corresponds to a single DNA molecule. Okay, so let's look at the different models that explain their ultrastructure. Okay, when I use the word ultrastructure, no, like when we say ultrastructure of bacterial cell, virus cell, it means the structure when we look in the microscope, okay? When we look in the ultra small magnified, magnification. Okay, ultrastructure means the cellular details, okay? Now the fiber folded model, it was proposed by Dupuy and is accepted widely. Then chromosomes are made up of chromatin fiber about 230 angstrom diameter. Each chromatin has only one double helix of DNA, which is in a coiled state, okay? This DNA coil is coated with histone and non-histone protein. Okay, so here the fibers are 239 angstrom in diameter. Then there's only one DNA double helix in a chromatin fiber and is coated with histone and non-histone proteins. So thus 230 angstrom chromatin fiber is produced by coiling of a single DNA double helix. The coils are stabilized by divalent cations like calcium and magnesium. So the DNA, it replicates during interphase and produces two sister chromatin fibers. The chromatin undergoes replication in the centromeric region also, so that the sister chromatin fibers are separated in this region also. The centromere also gets replicated so that you can separate from here, okay, right from the centromere. Now, I've already told you, when we fold the chromatin fibers, it reduces their length drastically, okay, and it's more more stable and more thick, okay, so it increases stability and their thickness. So it undergoes supercoiling, which further increases the thickness of the chromosome, okay. So not only is it coiling this way, it will supercoil, okay, supercoil, it will be even more coiled. Okay, so let's see. So this is the centromere, it will replicate, okay, then it will undergo supercoiling, okay. So there, even the centromere is coiling. These strands, they are coiling also, and then they will undergo supercoiling, means this one again, this whole thing will coil. Here inside this is small coils, okay, and again it will undergo supercoiling, okay. So that is basically your fiber folded model. The organization of chromatin fibers, any model of chromatin fiber structure has to account for packaging of a very long DNA molecule into a unit length of fiber, production of very thick fibers from a very thin DNA molecule, and beads on a string ultrastructure of chromatin fiber observed during replication. So how they organize is that, so number one, I told you DNA is very long, okay, from here to the moon, if you stretch it. So you have to pack a very long DNA molecule into a very short unit, okay. And the fibers have to be very thick, okay. We have to produce thick fibers from very thin DNA molecule. and then we have to arrange in that beads on a string ultrastructure. Okay, so this is your basic organization. Very long in a small unit. From thin fiber we have to produce something thick, and then beads on a string ultrastructure by coiling. Okay, so the coiled DNA model again, it was given by Dupuy. The single DNA molecule on a chromatin fiber is coiled in a manner similar to wire on a string. Okay, the coil will be held together by histone bridges produced by binding histone molecules in the large group of DNA molecule. Such a coil structure would be stabilized as a single histone molecule would bind to several coils of DNA. So it is just like you are coiling wires on a spring. Okay, then these coil will be held together by histone. Okay, and this histone will act like a bridge. Okay, so this is a simple explanation. So you remember from here, wire on a spring joined together by histone which act as bridge. Okay, so let's move on. Then the coil structure is coated with chromosomal protein to yield the basic structure of chromatin fiber, which may undergo supercoiling to produce type B fiber. Okay, so okay, again spring, no, histone bridge, then there is something coiled, okay, by some protein, okay, to produce a type A. Then again this type A will coil in itself to produce a type B. Okay, so this is the diagram, the DNA double helix, coil, and these are your histones, these lines which act as bridges. What theory? These act as bridges, these lines. Then you have a coating, okay, of a chromatin fiber. So this one is the fiber fold A. Okay, then this coating again it will coil and you will get a fiber fold B. So there. Okay, so again I'm repeating, nothing beats practicing with pen and paper. You will be fast, you will be neat. Okay, if you keep on writing and practicing your answers, you will be quick and neat.